Welcome friends to a new session on molecular biology. This session will describe the various DNA regions involved in transcription and transcriptional regulation such as promoters, enhancers, silencers and cofactors. Regulatory regions in DNA. Promoters, enhancers, silencers and cofactors are various DNA regions involved in transcription and transcriptional regulation. Promoter. A promoter is a region of DNA that initiates transcription of a particular gene. Promoters are located near the transcription start sites of genes on the same strand and upstream on the DNA, that is towards the 5' region of the sense strand. Promoters can be about 100 to 1000 base pair long, approximately 250 base pair upstream of the start site. There are two types of promoter elements, core promoter and proximal promoter. Core promoter is the minimal portion of the promoter required to properly initiate transcription. It includes the transcription start site, a binding site for RNA polymerase and general transcription factor binding sites, example Tata box. Proximal promoter is the promoter sequence which contains 250 base pair upstream of the start site of the gene that tends to contain primary regulatory elements and specific transcription factor binding sites. Distal promoter is the promoter sequence upstream of the gene that contains additional regulatory elements and specific transcription factor binding sites, often with a weaker influence than the proximal promoter. Prokaryotic and eukaryotic promoters. First, let us see about prokaryotic promoters. The sigma subunit of RNA polymerase mediates its binding to promoters in DNA. Hundreds of Escherichia coli promoters have been sequenced and found to have two short sequences within these promoters which are highly conserved to be recognized. These promoter sequences occur at about 10 and 35 nucleotide pairs upstream from the transcription start site. Thus, they are called -10 sequence or the -10 element or the Pribnow box and the -35 element or -35 sequence respectively. These sequences vary slightly from gene to gene and some nucleotides are highly conserved. The nucleotide sequences that are present in highly conserved region are called consensus sequences. The -10 consensus sequence in the non-template strand is TATAAT. The -35 consensus sequence is TTGACA. The sigma subunit initially recognizes and binds to the -35 sequence. Thus, this sequence is called recognition sequence. The optimal spacing between the -35 and -10 sequences is 17 base pair. Let us see about eukaryotic promoters. Eukaryotic RNA polymerase cannot initiate transcription by them. All three eukaryotic RNA polymerases require the assistance of transcription factors to synthesize an RNA chain. The initiation of transcription involves the formation of a locally unwound segment of DNA. It involves the interaction of several transcription factors with specific cis acting sequences in the promoter for the transcription unit. The promoters recognized by the RNA polymerase II consists of short conserved elements located upstream from the transcription start site. The conserved element closest to the transcription start site is called Tata box. It has a consensus sequence TATAAA and is centered at about -30 position. The Tata box plays an important role in positioning the transcription start point. The second conserved element is called the CAAT box near the -80 position and has the consensus sequence GGCCAATCT. Two other conserved elements, the GC box, where the consensus sequence is GGGCGG, and the octamer box, where the consensus sequence is ATTTGCAT, are often present in RNA polymerase II promoters. The CAAT, GC and the octamer sequences influences the efficiency of a promoter in transcription initiation. All active promoters contain at least one of these elements, but all the three elements are not present in every promoter. Gene promoters are typically located upstream of the gene and can have regulatory elements several kilobases away from the transcriptional start site, that is enhancers. In eukaryotes, the transcriptional complex can cause the DNA to bend back on itself, which allows for placement of regulatory sequences far from the actual transcription site. The RNA polymerase II dependent promoters in eukaryotes contain a Tata element which is recognized by the general transcription factor Tata binding protein, that is TBP, and a B recognition element BRE, which is recognized by the general transcription factor TF2B. The Tata element and BRE are located close to the transcriptional start site, typically within 30 to 40 base pair. Eukaryotic promoter regulatory sequences typically bind transcription factors that are involved in the formation of the transcriptional complex. An example is the E box, wherein the sequence is CACGTG, which binds transcription factors in the basic helix loop helix family. Example, ARNTL clock, that is aryl hydrocarbon receptor nuclear translocator like protein 1, circadian locomotor output cycles kaput. And another one is CMYC, that is mic oncogene. Some promoters in eukaryotes are targeted by multiple transcription factors which might achieve a hyperactive state leading to increased transcriptional activity. Bidirectional promoters. Bidirectional promoters are short, less than 1 kilobase pair, intergenic regions of the DNA between the 5' ends of the genes in a bidirectional gene pair. A bidirectional gene pair refers to two adjacent genes coded on opposite strand with their 5' ends oriented toward one another. The two genes are often functionally related, and modification of their shared promoter region allows them to be co-regulated and thus co-expressed. Bidirectional promoters are a common feature of mammalian genomes. About 11% of human genes are bidirectionally paired. Bidirectionally paired genes in the gene ontology database shared at least one database assigned functionally category with their partners 47% of the time. Microarray analysis has shown bidirectionally paired genes to be co-expressed to a higher degree than random genes or neighboring unidirectional genes. Although co-expression does not necessarily indicate co-regulation, methylation of bidirectional promoter regions has been shown to downregulate both genes and demethylation to upregulate both genes. There are exceptions to this. In some cases, around 11%, only one gene of a bidirectional pair is expressed. In these cases, the promoter sequence is implicated in suppression of the non-expressed gene. The mechanism behind this could be competition for the same polymerase enzyme or chromatin modification. Divergent transcription could shift nucleosomes to upregulate transcription of one gene or remove bound transcription factors to downregulate transcription of one gene. Some functional classes of genes are more likely to be bidirectionally paired than others. Genes implicated in DNA repair are 5 times more likely to be regulated by bidirectional promoters than by unidirectional promoters. For example, chaperone proteins are 3 times more likely to be regulated by bidirectional promoters, and mitochondrial genes are more than twice to be regulated by bidirectional promoters than by unidirectional promoters. Many cellular metabolic genes and basic housekeeping genes are regulated by bidirectional promoters. The overrepresentation of bidirectionally paired DNA repair genes associates these promoters with cancer. 45% of the human somatic oncogenes seem to be significantly regulated by bidirectional promoters more than non-cancer causing genes. Certain sequence characteristics have been observed in bidirectional promoters, like the lack of Tata boxes. But CAAT boxes are common, as they are in many promoters that lack Tata boxes. Although the term bidirectional promoter refers specifically to promoter regions of mRNA encoding genes, luciferase assays have shown that over half of human genes do not have a strong directional bias. Research suggests that non-coding RNAs are frequently associated with the promoter regions of mRNA encoding genes. It has been hypothesized that the recruitment of the enzyme RNA polymerase II and initiation of transcription usually begins bidirectionally, but divergent transcription is halted at a checkpoint later during elongation. Possible mechanisms behind this regulation include sequences in the promoter region, chromatin modification, and the spatial orientation of the DNA. A subgenomic promoter is a promoter added to a virus for a specific heterologous gene, resulting in the formation of mRNA for that gene alone. Enhancer. Enhancer is the DNA element that can enhance transcription. It may be located either upstream or downstream of the transcriptional initiation site. However, most of them are located upstream. In prokaryotes, enhancers are quite close to the promoter, but eukaryotic enhancers could be far from the promoter. An enhancer is a short sequence ranging from 50 to 1500 base pair or region of DNA that can be bound by proteins, that is activators, to activate transcription of a gene. These proteins are usually referred to as transcription factors. Enhancers are generally cis-acting, located up to 1 mega base pair away from the gene and can be upstream or downstream from the transcription start site and either in the forward or backward direction. Enhancers work as cis regulatory elements to mediate both spatial and temporal control of development by turning on transcription in specific cells and or repressing it in other cells. Thus, the particular combination of transcription factors and other DNA binding proteins in a developing tissue controls which genes will be expressed in that tissue. Enhancers allow the same gene to be used in diverse processes in space and time. There are hundreds of thousands of enhancers in the human genome. Furthermore, an enhancer does not need to be located near the transcription initiation site to affect transcription, as Some have been found located in several 100,000 base pairs upstream or downstream of the transcription start site. Enhancers do not act on the promoter region itself, but are bound by activator proteins. These activator proteins interact with the mediator complex, which recruits the enzyme RNA polymerase II and the general transcription factors, that is GTFs, which then begin transcribing the genes. Enhancers can also be found within introns. An enhancer's orientation may even be reversed without affecting its function. Additionally, an enhancer may be excised and inserted elsewhere in the gene and affect the transcription. That is one reason that introns polymorphisms may have effects although they are not translated. Silencer. A silencer is a DNA sequence capable of binding transcription factors. It is also called repressors. DNA sequence contains genes and provides the template to produce mRNA. This mRNA is then translated into proteins that activate or inactivate gene expression. When a repressor protein binds to the silencer region of DNA, RNA polymerase is prevented from binding to the promoter region. When the transcription of DNA into RNA is blocked, the translation of RNA into proteins is impossible. Thus, silencers prevent genes from being expressed as proteins. RNA polymerase transcribes the DNA sequences in the 3' to 5' direction, while the complementary RNA is synthesized in the 5' to 3' direction. An important region found in RNA is the 3' untranslated region for the activity of gene repression and expression. This is a region on the 3' terminus of RNA that will not be translated to protein, but includes many regulatory regions. There are many positions in which a silencer element can be located in DNA. The most common position of a silencer is found upstream of the target gene, where it can help repress the gene transcription. This distance can vary greatly between approximately minus 20 base pair to minus 2000 base pair upstream of a gene. Certain silencers can be found downstream of the promoter region located within the exon or intron of the gene itself. Silencers have also been found within the 3' untranslated region, that is 3' UTR of mRNA. There are two main types of silencers in DNA, the classical silencer element and the non-classical negative regulatory element. In classical silencers, the gene is actively repressed by the silencer element, mostly by interfering with the general transcription factor GTF assembly. Negative regulatory elements passively repress the gene by inhibiting other elements that are upstream of the gene. There are certain silencers in NREs which are orientation dependent, meaning that the binding factor binds in a particular direction relative to other sequences. Promoter dependent silencers are position and orientation dependent, but must also use a promoter specific factor. Another group of elements called polycomb group response elements, in short called as PREs, can allow and inhibit repression depending on the protein bound to it and presence of non-coding transcription. Silencer elements target the assembly of GTFs necessary for transcription of the gene. These silencer elements are mostly located upstream of the gene and can vary between short and long distances. For long range silencers, it has been observed that the DNA will form a loop in order to bring the silencer close to the promoter and loop out the interfering DNA. Silencer elements also target helicase sites in the DNA that are rich in adenine and thymine and are prone to unwinding and thus the DNA allows transcription initiation. The inhibited helicase activity leads to the inhibition of transcription. This is commonly seen in the human thyrotropin beta gene promoter. Cofactors. The discovery of transcription factors and their cofactors are important for transcription regulation. These transcription factors and their cofactors bind the DNA segments to activate or repress the gene expression nearby. Cofactors are often required to transmit regulatory signals between gene specific activators and the general transcription machinery. There are three types of general cofactors, such as 1. TBP, that is Tata binding proteins associated factors, 2. mediator complexes, and 3. upstream stimulatory activity derived positive cofactors such as PC1, PC2, PC3 and PC4, and negative cofactor 1, which is known as NC1, normally function independently or in combination to fine tune the promoter activity in a gene specific or cell type specific manner. Cofactors are required for the transmission of regulatory signals between the gene specific activators and general transcription machinery. Tata binding protein, TBP. The TBP is a general transcription factor that binds specifically to a DNA sequence called the Tata box. This DNA sequence is found about 30 base pair upstream of the transcription start site in some eukaryotic gene promoters. It is estimated that only 10 to 20% of human promoters have Tata boxes. Tata binding proteins associated factors is a part of TF2D multimeric protein complex that in turn makes up part of the RNA polymerase II pre-initiation complex. The TBP is an unusual protein which contains long string of glutamines in the N terminus of the protein, which binds the minor groove using a beta sheet. It also modulates the DNA binding activity of the C terminus, and modulation of DNA binding affects the rate of transcription complex formation and transcription initiation. The first Tata binding protein associated factors, that is TAF, was discovered in Drosophila melanogaster and humans. These proteins were identified as biochemically stable compounds and distinguished by a number which indicates their predicted or apparent molecular weight. TAF proteins adopt a histone like fold. TBP is probably not the only protein involved in positioning RNA polymerase II, but it is involved in double strand separation of DNA known as DNA melting by bending the DNA at 80 degree. The AT rich sequence to which it binds facilitates easy melting. Transcription coactivator mediator complexes. The transcription coactivator mediator complex of the transcription mediated by RNA polymerase II bridges transcriptional activators, coactivators, general transcription factors and RNA polymerase II to repress or enhance the formation of the transcription pre-initiation complex. It also participates in the formation of several functional complexes such as the activator recruited cofactor, cluster recruited to nuclear receptors and stimulator of RNA binding MET cofactor complexes that particularly works for activation of transcription by hepatocyte nuclear factor 4, another SRB MET cofactor complex, NAT complex that repress the activation of transcription by RNA polymerase II, cofactor required for specificity protein 1 transcriptional activation that is required together with TAF2s to activate the transcription factor specificity protein 1 and positive cofactor complex 2. Mediator complex subunit 1 is a component of mediator that associates with the nuclear hormone receptors. With the mediator kinase molecule, mediator forms the thyroid hormone receptor associated protein complex that repress transcription. With the subunit MED26, the mediator complex known as positive cofactor 2 activates transcription. Upstream stimulatory activity derived positive and negative cofactors. The upstream stimulatory factors are homo or heterodimers that pertain to the basic helix loop helix leucine zipper class of transcription factors to bind the E box element. Positive cofactors 1 corresponds to PARP1, that is poly ADP ribose polymerase 1. Positive cofactors 2 with MED26 to mediator, positive cofactor 3 to DNA topoisomerase 1. The negative cofactor 1 NC1 corresponds to HMGB1, that is high mobility group box 1 protein. In addition, other cofactors such as TAF1, BTAF1 and negative cofactor 2 can also modulate TBP or TF2D binding to the core promoter. Now, let us summarize the following discussion on promoters, enhancers, silencers and cofactors in transcription. Promoters, enhancers, silencers and cofactors are various DNA regions involved in transcription and transcriptional regulation. A promoter is a region of DNA that initiates transcription of a particular gene. Promoters are located near the transcription start site of genes on the same strand and upstream on the DNA. It is about 100 to 1000 base pair long, approximately 250 base pair upstream of the start site. There are two types of promoter elements, core promoter and proximal promoter. The promoter sequences in prokaryotes consist of two short sequences at -10 and -35 positions upstream from the transcription start site. Many eukaryotic promoters contain a Tata box, which in turn binds a Tata binding protein which assists in the formation of the RNA polymerase transcriptional complex. An enhancer is a short sequence consisting of 50 to 1500 base pair or region of DNA that can be bound by proteins, that is activators, to activate transcription of a gene. A silencer is a DNA sequence capable of binding transcription factors, that is repressors. Cofactors are often required to transmit regulatory signals between gene specific activators and the general transcription machinery. There are three types of general cofactors such as TBP, Tata binding proteins associated factors, 2 mediator complexes and 3 upstream stimulatory activity derived positive cofactors 1 to 4 and negative cofactor 1. Now, let me give you certain assignments based on the discussions what we had. 1. Write short notes on Tata binding protein, that is TBP. 2. Write short notes on prokaryotic and eukaryotic promoter. 3. Write short notes on bidirectional promoters. 4. Differentiate proximal and distal promoters. 5. Give a brief account on the two main type of silencers in DNA. I will also give you certain references for your further reading. 1. Book entitled Biochemistry 5th edition, authored by Berg, Timosco, Stryer in the year 2008, published by Freeman and Company, New York, USA. A second book is with the title Cell Biology, authored by Karp in 2014. It is a 7th edition, published by John Wiley and Sons, Singapore. A third book is with the title Molecular Biology of the Cell, 5th edition, authored by Alberts, Johnson, Lewis, Raff, Roberts and Walter in the year 2007, published by Garland Science, New York and London. A fourth book is with the title Gene 9, authored by Levin, 2007, published by Jones and Bartlett Publishers, Sudbury, Massachusetts. Fifth book is with the title Biochemistry. The authors are Davidson and Sitman, 1997, published by Waverley, New Delhi. Sixth is book with the title Principles of Genetics, authored by Gardner and Nestor in 1984, published by John Wiley, New York. Seventh book is with the title Principles of Genetics, authored by Sinnett, Dunn and Dobzhansky in the year 1958, published by McGraw-Hill, New York. I will also give you certain websites for your further reference. First one is www.bioencyclopedia.org. Second one is www.oxfordgenetics.com. We have come to the end of this session. We will meet in another session with a new topic. Until then, bye.